Our next speaker is Seong-Gi Kim from the Institute of Basic Science in South Korea. And he's going to share his most recent work in optogenetic silencing to explore the neural basis of resting state FC.
Thank you, Lauren and Lisa, and then also Sharon, to put together a great workshop, symposium together. Let me see, so I'm just figure out how to use it there. This is works. Han Seok-Moon, so is my postdoc. He cannot make this time, so I present his work today. And also Elizabeth gave a very beautiful talk just before me, and she actually gave what is the basis about the functional connectivity. So I'm going to talk about this extension about the whole talk. That is. So we all know Barack is here. So it's basically functional connectivity used routinely for the resting state fMRI analysis there. And this is a hemodynamic response, as all we know, there was one. And what we see, there's a lot of times homeotopic functional connectivity. So both hemisphere, same brain region area respond together. So question is, what is this is a source of this one? When you look at the calcium activity, also when you look at the hemodynamic response, they are well correlated. So it's appeared to be they have somehow neural basis about this bilateral functional connectivity to those one. When you look at what is the idea? So when you have a correlate together, so one idea is there is somehow anatomical connection between two hemispheres through the corpus callosum. A lot of research show that is not the case to those one. So when you look at the data set, when you see there is a resting state fMRI, I'll show this one, resting state fMRI here. This is a structural connectivity by the Allen Institute. This is all the metrics you see here. This is an ipsiloid side and controller side. Ipsiloid side, there is a very good correlation, but controller side, there is a lot of differences there. So there is no direct connection between these two hemispheres. Question is, what is the exact source of this neural source of this functional connectivity? One simple idea is they are common source. They are both hemispheres, they are correlated together, they are common source, project some information together. There is a lot of information about the basal brain, locus radius, there was one. Then another possibility, you have polysynaptic interaction. Somehow goes through another pathway. Question is how we dissect these two ideas. We know there is a possibility, but we cannot test that very easily. One way you can do it, you can manipulate the area. So for example, if you area A and B, there's two different hemispheres, A you suppress, silencing. So you have a baseline functional connectivity activity there, you're silencing those one. Then you have a signal decrease in this area. Then we have common input. Then the other hemisphere, there is no change whatsoever. Okay? Then if you have polysynaptic connection, then what you expect to have, there is a reduced activity in this area. So when you silencing certain part of the brain, then you can separate these two components. So this is the idea of what we have it. So how can you do it? One is we have a lot of different brain area. We want to selectively silence a lot of different areas. So we actually want to develop the optogenic manipulation with the pattern optogenetics. What is this idea is? Idea is we use the DMD chips, the digital micrometer device. You can turn it off on, so you can have a selectively silencing certain part of the brain. We implement this idea into the MRI scanner. So this, for example, you can see it here. This is a laser coming in there, DMD chip. The light coming into dichronometer, goes through those fibers, and project into the magnet. Then we got the response back, signal, then we can actually getting also is optical images. So you can actually do the pattern optogenic stimulation and optical imaging and fMRI together. So this is a simple cradle. There's a Bruker cradle here. We actually built this part here. This is a system. If you expand, you can see like this one. This is what you see the mouse is here. There was this star sign we project into the ears. So we can actually use this cradle, XYZ coordinate, you move around, we can focus onto the brain. So then we need a correlation between optical system and MRI system. So this is one is a challenging system. There we need to penetrate light into the cortical area. So if, for example, this is a mouse brain, and then you have cinder skulls. So we can have a light penetrate into the cortical area. Then we use this one is AGA solution with the detour. So we are trying to minimize MRI signal from this one, because otherwise there is too bright in this one. Then we also use the reference here. This is also MR comparable. So you see the surface core right on here. So then we can do, so this MRI image, we actually got a beautiful MRI images. We can actually do, then we can combine this MRI data set. These are all the 3 reference scan. These are optical images. We have, this is fluorescence image. We can actually correlate this together. So we can actually stimulate. So one actual question is always coming down to what is the difference between this one and this fiber optics stimulation. These are fiber bundles. These are depths. This Monte Carlo stimulation study there, you can stimulate, usually have very focal stimulation. Then when you do the pattern optogenetic, much larger area, low NA, much deeper stimulation, you can actually see here, this is a Monte Carlo simulation. You can see star sign going to all the way down. So this is an actual data set. You can show this is a light, this is where is a light signal coming in there. You look at the depths, much deeper penetration. So its idea is that this system is working. We want to implement this idea into the magnet to examine resting state fMRI signals. So what do we do there? We implement this one. We actually use 6 different target area. 6 different target area. This is we activate inhibitory cells. That means we have a silencing excited cell. So that's what we have a model system here. Then we do the exactly same experiment, kind of experiment in the resting state fMRI at 15.2. So we want to try to combine this data together. So let's look at the data set. This is, we actually stimulate M1. We actually silence the M1. Okay, this is the M1 area. So you can see the shape is what the M1 shape here. This we anatomically designed this pattern with silencing this area. This bluish area is all the negative change due to this silencing. You can see there is a lot of negative area. Well, you look at the area here, striatum. There is Elizabeth actually showed the striatum there. So you see a negative change, ATN, all different areas you have a negative change. Strength of this negative change is considered connectivity strength. You have a more connection, that means more changes. There's a smaller connection, there's smaller change. So we actually use this idea to determine strengths of spontaneous effective connectivity. So we did 6 different brain area we stimulate. This is all the stimulation site in the actually same animal. So you say it's not a different group. So you get the same animal, different stimulation site. We actually got a map it there. So what you see that's actually interesting there, you actually see the most like bilateral negative change. Okay. So then we can compare with the resting state fMRI data set. Top one is we do the 15.2 Tesla study. We do resting state fMRI. You can see there is the bilateral activity here. This is effective connectivity. Just we do the pattern optogenetic study. You can see they are quite similar. You can see the shape is coming down here, similarity there. All right? So you can actually compare these 2 data together. So this is if we look at this, it's just bilateral connectivity. Look at this one. This is a seed point. You can like ipsilateral side versus a contralateral side. So you look at this one. This is a ipsilateral side, contralateral side. If you got a functional connectivity, there is a bilateral connectivity. You can see very easily. All right. When you look at structural connectivity, that's what we had coming from Allen Institute, Atlas there. There's mostly like ipsilateral connection. When you look at the spontaneous effective connectivity, just with the silencing data set, we can see there is there's a bilateral connectivity coming down there. They are quite similar between these 2. If you plot together, this is a FC strength from the resting state fMRI. This is a strength of optogenetic fMRI signal changes. You can see they are quite good correlation. So we can actually conclude, based on this measurement, okay, spontaneous neural interaction is a primary drive of a resting state functional connectivity. All right, that's just a good sign. So next question we have it, how many connections do we need it? Okay, we know there is no direct connection is between two hemispheres. So we have to figure out how many synaptic connections are needed. So we actually going to the Allen Institute. It's a projection matrix there. So you can actually go into, this is a matrix from the all different area. It's a cross connection. Then what we actually simulate, we have a six target we used. Then we have all different target area. Then we making first connection, second connection, third connection, whatever. We can actually simulate all of this connection based on the Allen Institute data set. So this can be done very easily. We can combine this one, then we figure out how many connections do we need. Just a simple linear is a basically combination. This is experimental data set, then first order, second order, third order, etc. Those. All right, then we can figure out how many we do it. And what we found out there, we only need three connections. So this is coefficient. You have a first connection is pretty high. It's about 60%. Then you got a second connection, third connection. Then we probably do not need to have a fourth connection. So it's based on this one, we actually need to have spontaneous activity require at least 3 synaptic connections. Then we can actually explain our bilateral activities. It's in conclusion, fMRI pattern in response to pattern optogenetic silencing is very similar to resting state functional connectivity. Bilateral functional connectivity can be explained by 3 synaptic connections at the resting state condition. We do not know how much difference between polysynaptic connection, obviously there is a polysynaptic connection, and also common input. So we did not investigate these 2 things there, but there can be, both exist. Thank you for your attention. Okay, I saved my time. Thank you.
We have time for questions.
I have a lot of questions. There are a lot of time left. I could not believe I actually finished earlier than what is my allocate time.
Terrific talk, Seong-Gi. So, not so sure about the mouse visual cortex, but I know primate visual cortex pretty well.
Yeah.
So, the geniculate, which is the thalamic nucleus, basically, goes to both sides, but in different fractions. So is that an origin point for what you see? And did you try, I mean, it's fairly deep, so I don't know whether you can stimulate it with optogenetics, but is that a way to explain the connectivity and the difference between left and right?
I think there's probably, I don't know exactly where, we don't know exactly what the source of the synaptic connection. So presumably we actually think it's going to a thalamus. Thalamic somehow connection going to there. So that's what your question is? Yeah. So we don't know exact origin, but that's our guess. Guess is more like it. So next step we need to have, we actually manipulate thalamic nuclei. So what we need to have a next step going to chemogenetically silencing to the, when you do this type of experiment, we silencing certain part of the thalamic nuclei where we can break this connection.
Yeah, so dreads in the geniculate might be.
Yeah, so we need to have for example, dread and silencing these 2 combined together. Somehow we need, then we can break it. We need to break this connection. Otherwise, we don't know where is the exact pathways. We know it's a 3 connection. And we are guessing it's a thalamus, but there can be other place too.
I have a question about when you silence those neurons, what type of neurons? Because when you have different levels of connections, there could be different population of neurons connected, right? So when you say thalamus and cortex, there will be different type and cortical.
So what we use is called the VGAP mouse. VGAP mouse is a channel adoption in inhibitory neuron. So we stimulate is optogenetically. That means activate all the inhibitory neuron. then silencing all excited neurons. This is all up to the all six layers, because this optogenic stimulation actually penetrates going to the sixth layer, and we actually record activity. I didn't show any neural data set, but we actually see the silencing all the six layers. So that means what we are seeing is all projection from sixth layer in the excited neuron, that all the downstream activity. So we don't know which layer. Only thing we know, down.
We still have time?
We still have time.
I see a question back here.
I mean, there was a shining here, so it's hard to see that.
Thank you very much for the talk. It's really inspiring stuff. Can you just maybe go back to the conclusion of most of your functional connectivity results can be described by 3 synaptic connections? Because when I see your plot, I see, really the vast majority seem to be described by a single, like monosynaptic connections. Then of course the correlation goes down. So are there other measures that you use to kind of examine the variance explained, for example, or something like that, based on the number of connections that you examined?
So it's a lot of things going to a single monosynaptic connection. Okay, true, all right? But that's not sufficient enough to explain everything there. So we need to have 3 synaptic connections. So it's a monosynaptic connection explain a lot of those one, especially like epsilon size. Whenever you stimulate the one, then we look at all epsilon size, you can explain by monosynaptic connection. And problem is when you do the contrary side, it's like the other hemisphere. The connection is not easy to explain by monosynaptic connection. So we almost need to have not a 2 and almost a 3 synaptic connection. Some of is area. Some area is probably monosynaptic connection can be also explainable. Did that answer your question?
Great. Okay. One.
Just to follow up with that, how heterogeneous is it across the brain?
Pardon me?
Is there like a profile of heterogeneity of the number of synaptic connections that you found?
I don't know whether I understand your questions properly. So
Because you're saying on average it's about 3 flow synaptic.
yeah.
How heterogeneous is it across the entire brain?
How heterogeneous in the sense? Okay. So it's really I didn't do all the experiment, so I cannot. I don't know my answer is correct. So it's when we do single synaptic connection, we can explain a lot in the epsilon side. Okay. When you then you go to more and more connection, there is a more explainable to the bilateral connection. So essentially, it's like when just we look at simulation, we are nothing to do any fMRI data set. Just look at simulation. We go to 6 connection. We can explain everything in the sense. All the bilateral connection beautifully shown. So it's essentially it's like we cannot say only 3 connection. Could be more than 3 connection. Based on our data set, 3 connection is sufficient enough, but could we need to have a more connection too. So it's not easy to say how many % like that one. And I think it's like a polysynaptic connection can explain about resting state fMRI data. That's what I actually want to say there. I don't know if it answer your question properly or not. That is, we can talk later.
Great. Okay. Thank you again, Sanjay.
Thank you. Yep.